First one is isotonic. This is isotonic. This one is where the cell will shrink. This is hypotonic. And this is hypotonic, and this is hypertonic, excuse me. The second one there is hypertonic, and the third one is hypotonic. It grows, it gets thicker. Okay. So, sodium-potassium pump. Does anybody remember this from biology? Sodium-potassium pump. What is that?
I kind of remember it, but I don't know.
Kind of remember it. So, who had Marie? Cammie did. Cammie should know. Do you remember? What does it sound like? Requires energy. Okay. I'll go with that. Sodium and potassium. These are the chemicals, and we're not going to go into it very deep here, but sodium potassium pump is what actually makes it so our nerves work and keeps our cells with a certain electrical charge. They all have an electrical charge. Remember in the Matrix with Keanu Reeves? They stuck those things in their brain and took their energy off of them. That's kind of what we do. We have an energy, and that's what makes that energy. That movement of cells back and forth will create that electrical charge that goes down the nerves. It makes our cells have a certain electrical charge too. We have a negative millivolts, negative 70 millivolts for our cells. That's the resting membrane. And we exchange the sodium and potassium, and it flip-flops that electrical charge, and we get a different impulse, a different charge in that cell. So that's the sodium-potassium pump. It moves the sodium and potassium in and out of the cell. Fuses out, pump it back in. Okay, that's the pump. Keeps us alive. Very important little mechanism that we have to have in order for us to work. And it also affects the proton pump, and when we're talking about energy production. Now, active transport, I have two types. We have active transport and vesicular transport. Both use energy, and it requires the carrier proteins to pump the solution or the solute into the cell. So we're actively transporting substances into the cell. It takes energy. So grab it on the outside of the cell and bring it on the inside of the cell. Active transport uses energy, and we're taking things usually from a higher concentration against its gradient to an even higher concentration, or from an area of high concentration to an area of, from area of low concentration into an area of high concentration. We have to push it in. It's kind of like if you have this little car and you have a bunch of clowns, and you want to put a bunch of clowns in your car, you have to start pushing them in. You just pack them in there, keep pushing, pushing. That's kind of what we're looking at. We're trying to move them into areas of low concentration to areas of high concentration. It takes energy to do that. Now the proton pump this is the biggest thing with our production of ATP. In the electron transport system, the proton pump is what makes things work. That's what the exchange of those protons and electrons give us that ATP production. This is the sodium-potassium pump, and it takes energy, and we're taking the sodium and the potassium and exchanging it. You can see that on this one we have more potassium inside the cell, and we're going to move it to the outside of the cell. The sodium is on the outside, and we're going to bring it to the inside. We're exchanging them. And when we do this, it creates an electrical charge, a shock that goes down the nervous system. So we need this for our nerves and for our muscles to work properly. Without this little exchange of sodium and potassium, our muscles and our nerves don't work. We just sit there and stare into space. Nothing happens. So some of the sodium is diffusing out of the cells, and then we have to pump it back through. We have to go through these leakage channels back into the cells. Now I know that some of you have had this and some haven't, but this is a review that we'll talk about more in the nervous system. So kind of keep that in mind. We're not done with this yet. We'll continue and talk about it when we talk about the action potentials and how it becomes more important. But for right now, this is kind of a standalone thing, and it doesn't make a lot of sense. But when we get into the nervous system, it will all come into play, and you'll be able to understand it a little bit better. Okay? So it creates the charge, basically, on our cells. So resting membrane potential is the amount of the gradient, the electrical charge is established by this, and we're moving the potassium out of the cell, and we're going to put the sodium back in, and then when it exchanges is when we get that flip or that electrical charge. So some of the things that happens in the cell that are kind of important for the cells to function better. We have exocytosis, endocytosis, phagocytosis, and then we have those are what makes the cells so that they can function properly. Now I can see it better. So what is exocytosis? Anybody know? What's exo mean? Exit? That's a good way to look at it. Exit? Or exo? Outside, right? Exo is outside. How about endo then? Inside, yep. How about phagocytosis? Phage, this comes from phage, means to eat. So this is the cellular eating, phagocytosis. Penocytosis is cellular drinking. Peno is small, right? So those are what we look at with that. We use this with vesicular transport. A lot of you asked the other day, where is the vesicular area? Where are the vesicles at in the cells? Well, little vesicles are these little pouches, these little bubbles almost inside the cytoplasm that carry things back and forth in the cell. From the outside of the cell to the inside, and from the inside to the outside of the cell. That's vesicular transport. We use this to keep things going into the cell or out of the cell. So if the cell produces something, we have to get it out of the cell. If the cell needs something, nutrient, we have to bring it into the cell. So this requires energy to do this. Everything that we do requires energy, basically. We have exocytosis as transportation of a product outside of the cell. Endocytosis is bringing it into the cell. Transcytosis is transporting it across the cell, from one side of the cell to another side of the cell, and then out of the cell usually. Now the substances that we look at can be anywhere from a protein to a lipid that we're taking out. We can make a hormone and take it out of the cell by endocytosis. We could use the phagocytosis process to make us so that the cell eats, gets some glucose in it, or gets nutrients. Penocytosis is cellular drinking, so we're bringing water in large masses into the cell. So with phagocytosis, the cell is kind of, let's imagine that the cell is fairly round, kind of like your cheek cells yesterday, right? And it comes up to, let's say, a bacterium. We have a white blood cell that comes up to a bacterium, and we're not going to get specific on which ones does this, right? It's a big white blood cell. And it comes up, and it opens up, makes kind of an indentation in it, and it surrounds the bacterium and closes it around it. Munches it up, breaks it apart, destroys it, right? That's a really gross explanation of how it's eating. Not so much gross, it's just that's how it works on a cellular level. So it engulfs it, it kind of surrounds it. And these little white blood cells kind of crawl around your body, inside and outside, around your cells, inside and outside your tissues, looking for dead cells, different bacteria, viruses, pathogens that are there, parasites, right? We all have them. They're all there. 70% of the human population have parasites. It's kind of scary, right? But we do. So these little things are trying to eat them up as fast as they can. Pinocytosis is cellular drinking. They pinch off into vesicles, you surround some water, you pinch it off in vesicles, they keep that water inside of the cell and they can use it later. Find this in the small intestine a lot of times. So some of the things that we have hormone secretion, neurotransmitter release. This should be exocytosis. Mucus secretion and ejection of wastes are all exocytosis. So change that to exocytosis. Little spelling error there. So our cells will create hormones. Inside the male reproductive system, there's little cells called seminiferous cells that produce the testosterone that give us the possibility of producing sperm. Females have little cells that produce estrogen and progesterone. Those are produced, they didn't just pop up, they are produced by our cells. And the genetics that we have in our nucleus tells us how to produce those hormones, how to make that estrogen and progesterone and testosterone. It's all in the genetic composition inside of our genes. But the cells have to make them. So once we get the estrogen produced inside the cell, we put it in a vesicle, then we transport it to the outside of the cell, and we spit it out. We open up the cell membrane and release it into the area around the cells, and it goes then into the bloodstream, into every part of the body. Okay? So let's go over the organelles, the different organelles of the body. Somebody tell me what about the nucleus. What is in the nucleus, and what's the deal about the nucleoli or nucleus? What is the difference? What is it? What say you?
The nucleus is the control center, which contains the nuclei.
Okay. And what's inside there? What's inside the nucleus? DNA. That's our chromatid material. Our genetic material makes us what we are, who we are, how everything works in the body, inside there. So it's genetics. It's our library of everything that we need to have. Its response to signals. So the cell is sitting there and it gets a signal from the outside. It makes it do certain things. It gets a chemical that comes to the outside of the cell. We transport that chemical in. It stimulates the cell to produce something. We have this ability to make our own hormones, our own proteins, our own fats. But we have to have the genetic blueprint to make that happen. So inside here we have this nuclear material, our blueprint of life. And we have these little pores on the nuclear membrane. Surrounding the nucleus is a nuclear membrane, which is it's a bilayer just like on the outside of the cell. But it has little pores in it, little openings, that allows the genetic material to be copied and released outside into the cytoplasm. And then we also have what they call the rough ER, that is a continuation of this membrane around the nucleus. Same type of a tissue membrane. Now, we have to remember, they always look so big in the pictures, but these are so very, very tiny, you can't see them with our microscopes. You have to have an electron microscope to be able to see these structures. And they're very fuzzy. So imagine all this stuff happening in each one of your cells 24 hours a day, and you can't see what's going on. We have no clue, as far as us, no scientists have looked at it, but they don't know how this all works. They just kind of, oh, I guess that's how it works. They do research. Fantastic stuff. This is the kind of a round area inside the nucleus, and it has been found to to deal with the RNA synthesis. So ribose RNA, rRNA is ribosomal RNA, and by doing this, it produces a special RNA from the DNA that we have. So we take the DNA, we unzip it, we open it up, we make a copy of one side of that DNA called ribosomal RNA. We ship it outside of the cell into the cytoplasm where it can be combined with the ribosome, and it makes the functional units work better so that they work. Now chromatin is the DNA. That's what makes our DNA. You have histones, proteins, and RNAs 10%. This makes up our chromatin material, or our genetic material, basically.
Question. Is histones what the DNA wraps around?
What's that?
Is the histones what the DNA wraps around?
The histine's a protein, yeah, it gives it the backbone. Kind of gives it that structure. It's a large molecule that it attaches it electrically or chemically to it, so that it makes it more of a stable molecule. All right, so here are some of the organelles that we're going to talk about. Different ones. Let's go through each one of them as we go along. So here's the mitochondria. Mitochondria, or mitochondrion, as some of them say. It is a double membrane structure, just like our cell membrane. Our plasma membrane is a double layer membrane. Provides most of the ATP production. Now, in primitive cells, they don't have a mitochondrion. Most of the bacteria don't have any organelles at all, but they don't have a mitochondrion. They can produce a small amount of ATP in their cytoplasm. But when the mitochondrion came into view millions of years ago, probably about 1.5 billion years ago, these little mitochondrions came into the cell and started taking up residency there. Call it endosymbiosis. A symbiote is an organism that lives in conjunction. So if you're 35 years old and you're still living at your parents' house, you're a symbiote. Or a drag on their environment, I don't know which. But
It's the opposite. You're 35 years old and your parents have you.
could be that way too. They could be a symbiote too.
It is
As long as you're cohabitating and functioning properly. Now, if you're not, you become a parasite, and that becomes more of a situation. Right?
That's my dad.
These mitochondria contain their own DNA molecules and their own RNA molecules, which really is cool because you can take the mitochondrion and go back in time and trace your ancestry back millions of years because of the mitochondrial DNA. What's really cool is that when you have conception, you have the sperm and the egg come together. The sperm goes into the egg, but the mitochondrion isn't viable in the male. Only the mother's mitochondrion is passed on to the babies. So when you go to the Maury Povich show and you find out who's your daddy, they can use two different DNA. They can use the DNA in your nucleus, or they can use the DNA in the mitochondria. And that's two different tests. The mitochondrial DNA is actually more specific than the nuclear DNA. Okay? So keep that in mind when you go see Maury, or Jerry Springer, or somebody, I don't know. Ribosomes. These are basically little granular things that form the sites of protein synthesis. They help us to make proteins. They are bound. We have some of them that are attached to the endoplasmic reticulum, and it makes bumps on that, and some of them are just free-ranging. We have free ribosomes coursing throughout the cytoplasm, and then we have others that are attached to the endoplasmic reticulum, and they are sites for protein synthesis. They make our proteins. Now the rough ER is named rough ER because of the ribosomes that are basically attached to them, chemically attached to those little membranes. The endoplasmic reticulum is a membrane that is contiguous with the nuclear membrane. It's attached to it. Here they produce proteins and phospholipids, which is kind of interesting. You can have different types of substances being produced in different cells. The smooth endoplasmic reticulum is smooth because it doesn't have ribosomes on it. It doesn't have these little bumps. These little bumps are what makes the proteins. So they have different functions in different areas. In the liver, they help with the cholesterol metabolism. They break down the glycogen, and they're more functional that way. They help detoxify drugs. And they also synthesize steroid-based hormones, like testosterone and things like this, in different cells. In intestines, they're there for absorption and synthesis of fats. So you can make a fat from these endoplasmic reticulum, the smooth ones. And then in the skeletal muscle and the cardiac muscle, they have a lot to do with the calcium release. They make it so that we release calcium in the proper times, in the proper quantities, so that our muscles can contract. They make them actin and myosin molecules work better by supplying the calcium necessary for this reaction to take place. Now the Golgi bodies, or Golgi bodies, I've heard it both ways. These are the storage facilities. They're packaging areas of the body. So once the endoplasmic reticulum make the proteins or the fats, then we have to package them together and put them in a vesicle to ship them out, to get them out of the system. And that's what the Golgi bodies do. Whoops. There we go. Another organelle is called a lysosome. The lysosomes are pretty cool because they helps to break down substances that are in our cells that aren't supposed to be there. They break down, they help with breaking down the bone to release calcium. They destroy cells that are injured. They'll take out bacteria, viruses. They're a pouch of enzymes, lytic enzymes, that they find something inside the cell that's not right, maybe an organelle that needs to be destroyed, it's getting old. They'll go up, they'll allow them to come in, they'll combine it with the enzymes, and it destroys it, kind of basically dissolves those substances that make it into those cells. Peroxisomes, last one I'm going to talk about today. These are really kind of cool ones. Really, they're little pouches, they're little vesicles filled with hydrogen peroxide. So what do you use hydrogen peroxide for in the real world? What do you use it for?
To clean blood.
To clean your blood, clean your cuts, your boo-boos, your owies, right? What else? What else can you use hydrogen peroxide for? Your teeth, yeah. You brush your teeth with hydrogen peroxide toothpaste and whitens them up. What else?
Is that the stuff you put in your ear?
Yeah.
Ears?
Is that hydrogen? I always do these two things mixed up. If you like, get water in your ear, you can pour it in your ear.
Oil.
Oil? I don't think that's oil.
Yeah, you put baby oil in your ears and take the water right out.
That.
Yep.
Huh.
Don't put hydrogen peroxide in your ears, it'll hurt.
I always think you do. It kind of looks like bubbling and fizzing. Just don't get it in your eye. Then.
Baby oil, much easier on your end. Yeah.
Huh.
So hydrogen peroxide, really quick. The stuff you get at the store is about 1.5% to 3% hydrogen peroxide. You can, if you mix it, you can get food grade hydrogen peroxide that's 35% pure. If you mix that and make 10% hydrogen peroxide and you put it on your hair, it'll bleach your hair blonde. If you put it in stronger at 35%, it will burn your skin. It'll take a wart off immediately. Just start burning it off. It takes a couple times to do it. The wart will be gone. 50% hydrogen peroxide is what they use to send the space shuttle up into space. It's full of oxygen. And when it dissociates, you get hydrogen and oxygen, and it's very explosive, and that's what they sent it up into space for. All right? So, have a good weekend, work on your post tests, get all that stuff done.